This video will be on chapter 12, which is DNA replication. So DNA is replicated during S phase in the cell cycle, and the way it happens is in a semi-conservative manner. And so what that means is that the two strands of that double helix DNA actually separate from each other, and the bases where they would normally be hydrogen bonding, that those hydrogen bonds would break apart, and each of those old strands, which is shown in blue, is used as a template to make a new strand of DNA. So that's why it's called semi-conservative. When you finish up, you end up with one of the strands being the old DNA and the other strand being the new DNA. There's an experiment done by Meselson-Stahl which actually proved that DNA replication is semi-conservative in nature, and they did a really elegant experiment in which they marked whether that DNA was the old DNA or the new DNA by using different isotopes of nitrogen. We're going to talk more about this in class and actually consider what their results were, but they considered three possible ways that DNA could be replicated. Either conservative replication, in which one of the strands that result from DNA replication is all the old DNA and the other strand would be all new DNA, or dispersive replication, which the old and the new DNA are completely kind of mixed together, or semi-conservative replication in which one strand is old, the other one is new. Briefly, what Meselson-Stahl did was they used nitrogen-15, a heavy isotope of nitrogen, and they grew bacteria in that isotope for a long period of time, so all the old DNA was that heavy isotope. And then they switched the bacteria to a solution of N14 and allow that bacteria to grow exactly one time or exactly 2 times. And what they then did is took that DNA from those cells and separated it by centrifugation on a gradient. Just want to point out that in this ultracentrifugation step, you will separate out the DNA that has the N15 and the N14, but you can't actually separate the strands of DNA, the two parallel strands that are found in the double helix. Okay, so as I said, DNA is replicated in a semi-conservative manner, but there are different types of genomes. And so we've talked about both eukaryotic genomes and bacterial genomes. And I'm going to start out with a bacterial genome or prokaryotic. Bacterial genomes are circular, and so here we have this round piece of DNA, and there is one place where replication will begin, and that is called the origin of replication. And it is at this one place that you begin to separate the strands, and then you continue around making a new strand using the old strands as template. This is actually called theta replication, and it's called theta because that figure actually looks like the Greek letter theta. In contrast, in eukaryotes we have linear chromosomes, and there are many, many places, in fact thousands of places along the chromosomes where replication will begin. And so if you have a linear chromosome like this, what does not happen is we don't start at one end and proceed along that chromosome. We start in the middle at several origins of replication, and then we proceed from each of those origins going out in either direction. Eventually, those separate origins will join up together, and we will get 2 complete new strands of DNA. So DNA polymerase III in bacteria is the primary enzyme that is used for DNA replication. This is a large multi-subunit protein, but it has some really severe limitations that dictate how DNA replication happens. So these are some very important points to take note of. First of all, DNA polymerase III synthesizes new DNA strands in a 5' to 3' direction only. And secondly, it cannot start a new strand. It has to actually extend an existing strand of nucleotides. So that reaction is shown here. So here on the right would be the template strand, and the new strand that is being formed by DNA polymerase III is shown here. And so we have a new nucleotide coming in at the 3' end of this strand that is being synthesized. and it will attach onto that OH actually. So the phosphate group will attack that OH and form a bond with it. In the process, these two phosphates are cleaved off, and this actually provides some energy for the reaction. DNA polymerase could then bring in another base, this time it would be a G, and make another phosphodiester bond. Every time we start DNA replication, we're going to separate those two strands, and a replication fork will move in either direction from that beginning point. Now, it's very important to remember that it is the new strand of DNA that is synthesized in a 5' to 3' direction. And because of this limitation, there are going to be two ways that the strands in one particular fork are formed. One of the strands can form toward the replication fork, and this will be called the leading strand. This one will be synthesized continually as the two strands of DNA continue to separate and the replication fork moves to the left. In contrast, the other strand has to be synthesized away from the fork. And so this one, the new strand, is going to be anti-parallel to the existing template strand, and it's going to go away from the fork, and this one is called the lagging strand. On the other fork from one origin, the leading and the lagging strands are also found. They are just going to be opposite because they're going in the other direction. I expect you guys to be able to identify which strands are the leading strand and the lagging strand in a DNA replication fork. Next, we're going to talk about the 3 stages of DNA replication, which starts with initiation and unwinding, then elongation, and then termination. Some of these stages are different between prokaryotes and eukaryotes, and so I will highlight the differences between these types of organisms as we go through. So first of all, initiation and unwinding in prokaryotes. So how do we get started? As I said before, replication begins at the origin of replication, and this is called ORIC. There's only one of those per bacterial chromosome, and at that particular sequence, which is shown in green on this figure, there are a cluster of proteins that bind, and these proteins are called DNA A proteins. These particular proteins bind, and then they cause a region of the DNA right next to it to unwind. And this region right next to it is actually easier to break open because it's made up of primarily A's and T's, which only have 2 hydrogen bonds between them instead of 3. So once we get this small region of unwinding, then that region is bound by something called single-strand binding proteins, which stabilize that single-stranded structure. Once that opens up a little bit more, proteins called helicases can get in there at either fork and begin to separate the 2 strands of DNA at the hydrogen bonds in either direction. Another very important protein in prokaryotes are a class of proteins called topoisomerases. There's a number of different types of topoisomerases. One is named DNA gyrase. All of these proteins release the tension and the supercoiling on the DNA. So you can imagine that when you open up 2 strands of DNA, those twists in the helix have to go somewhere else. And so they actually end up going into the other part of the DNA that is still double-stranded, and that creates a lot of tension and it needs to be released. And so DNA gyrase makes these little cuts, and then the DNA rotates and tension is released, and then it anneals those DNA strands back together again. Initiation and unwinding in eukaryotes has the same rough idea, but a couple of different parts to it. So this has been studied pretty heavily in yeast, which is one of the simplest eukaryotes. And instead of that protein DNA A, there is a protein complex called ORC. Now ORC stands for Origin Recognition Complex, and ORC will bind to these particular sequences that are found at many different places along each linear chromosome. This consensus sequence is the binding site, and ORC will be the thing that actually initiates that replication. As we found in bacteria, right next to that origin is an AT-rich region, which will be the place where the unwinding of the 2 strands of DNA first occurs. There are a couple of more complex things going on in eukaryotes. One potential problem is that S phase is when DNA replication has to happen, and each of those origins has to be initiated only one time. So you could imagine that if there was no regulation here, that we could initiate an origin, start copying it, and then another ORC protein could bind to that origin that has already been copied and actually initiate replication a second time. There's a way to prevent that mistake, and that is done by something called replication licensing factors. So during G1 of the cell cycle, there are certain factors available. These are factors called CDC6 and CTD1. These are licensing factors. These factors are necessary in order for ORC to begin replication, and they are only available in G1, and so every origin during G1 gets bound by an ORC complex, gets licensed by these licensing factors, and then once the cell enters into S phase, all of the excess licensing factors will be degraded. So that ensures that each replication origin is only initiated one time. So once we get into S phase, same kind of thing happens. ORC, along with the licensing factors, causes these two strands of DNA to separate from each other. Helicase can then get in there and begin to separate the strands even more, and then eventually DNA polymerase can get recruited to those replication forks. The other factors, helicases, topoisomerases, single-stranded binding proteins, those are all in common between prokaryotes and eukaryotes in their initiation and unwinding. Next, we'll talk about elongation, first in prokaryotes. So this is an overview of the process of elongation starting from initiation. And so after initiation, we've separated those two strands at that origin, and helicase gets into either of those forks and begins to separate the two strands of DNA in either direction. At that point, DNA polymerase can get in there, and also another enzyme called primase. So if you recall, DNA polymerase 3 cannot start new strands of DNA. It has to add to an existing strand. And so the workaround solution for that problem is an enzyme called primase, which comes in and makes a little RNA primer complementary to each of those DNA template strands. Here we're going to make an RNA primer, it's shown in green, and this will then be a substrate for DNA polymerase 3 to come in and bind. This top one will be the leading strand being synthesized toward the fork, and in the other direction, at the other fork, a primer is made on this strand as well, and we get synthesis toward the fork. Primase also will make an RNA primer complementary to the DNA right near to the fork on the lagging strand, and this will act as a substrate for DNA polymerase 3 to bind and to begin to make a new strand of DNA going away from the fork. So here we have the formation of the lagging strand going away from the fork. This happens on both of these forks that are shown. So I'd like to just focus in on one of the replication forks and consider what's going to happen here. So here in blue is the old DNA and the new DNA is in yellow. First of all, primase will come in and make a RNA primer, and it will get that leading strand started. So the bottom strand in this particular figure is the leading strand. The leading strand will be synthesized continually, and as those two strands of DNA separate from each other, it will continue to be synthesized. In contrast, the lagging strand is going to be synthesized in a discontinuous manner. And so what will happen here is primase will synthesize a little RNA primer, and DNA polymerase will bind on and synthesize that piece of DNA. And then it will come back to another RNA primer and synthesize yet another piece of DNA from there. At this point, another enzyme will come in called DNA polymerase 1. DNA polymerase 1 will chew away those RNA primers, and DNA ligase then will come in and anneal those two pieces of DNA together. This discontinuous method of DNA synthesis produces these individual little pieces of DNA that are called Okazaki fragments. Each Okazaki fragment is somewhere between 1,000 and 2,000 nucleotides long in bacteria. In eukaryotes, however, these are much shorter. These Okazaki fragments are only about 100 to 200 bases long. A couple of other things I want to point out. So each replication fork actually has two DNA polymerase molecules attached to it, one for the leading strand and one for the lagging strand. And surprisingly, those two DNA polymerases are actually bound together. They link together, and how this happens then is that when the lagging strand is synthesizing away from the fork, it causes the DNA to kind of loop out in the process. DNA polymerase III also has a proofreading ability, so if a mismatch is formed, if the wrong base gets in there, it can actually slightly backtrack, remove that mistake, and continue synthesizing. This is really fast, 1,000 nucleotides per second. If we look at elongation in eukaryotes, it's really almost completely identical. There are a couple of very slight differences that I'll just highlight here. First of all, there are a lot more DNA polymerase enzymes, and in fact, they have much more specialized functions. So for example, there's a different DNA polymerase enzyme for the leading strand versus the lagging strand. We are not actually going to go into and learn those details about all the different types of DNA polymerases. We're just going to use the DNA polymerase names that are found in prokaryotes. The second major difference is that eukaryotes have nucleosomes on the DNA, and so this really complicates DNA replication. And what has to happen is those nucleosomes in which the DNA is wrapped up have to be removed prior to replication and then put back on after replication. After replication, we'll also need about twice as many nucleosomes. And so what happens here is there is a factor called CAF1, which actually assembles those nucleosomes, puts them back on the DNA after the new strand has been synthesized. Last step we'll talk about is termination. And termination in prokaryotes is actually really simple because you just come to the end, it's a circular chromosome, and the two DNA molecules just separate from each other. The normal machinery can just fix up that last little bit, which would require DNA polymerase I to remove that last RNA primer and DNA ligase to then seal those final strands. Termination in eukaryotes is a lot more complicated. So as we talked about before, we have multiple origins of replication, and these begin to join up, and it's really not a problem when we have two forks that then join together. Again, it's the same normal machinery that just fixes up that last little bit, removes the RNA primer, DNA ligase gets those last strands together. The problem arises actually at the ends of chromosomes, and so we're going to talk about that next. At the very end of a chromosome is a structure called the telomere, and the telomeres are problematic. So if we consider that here is a origin of replication, we've got forks going in either direction from it, and then one of those forks is going to get to the end of a chromosome. Let's just consider that this region on the right is the end of the chromosome, the absolute end. What's going to happen here is these two template strands will completely separate from each other. On the top one, where the leading strand is forming, there's no problem. It's just going to continue synthesizing DNA until it gets to the very end of the DNA molecule, at which point the entire complex just falls off and you've completely synthesized a new strand complementary to that template strand. The problem arises on the lagging strand. And so here we've got the Okazaki fragments, so we're forming these strands in a discontinuous manner. And at the very end, right near the end, we can make an RNA primer, we can make that last Okazaki fragment, but we've still got some RNA primer. RNA is not going to be maintained in a DNA genome, and so we're going to degrade that last bit of RNA, and it will leave this gap. At the very end, we'll have a little region where we just can't make a new strand of DNA that's complementary to the template strand, which is in blue. What will happen in this case is that single-stranded region will become degraded. The result of this is that every time a cell divides, undergoes S phase, a little bit of the ends of the chromosome will be lost. The amount of base pairs that are lost at the end of chromosomes is somewhere between 25 and 200 bases in each cell division. Now, that doesn't seem like that big of a deal. If you just lose a tiny bit at the end, we've got pretty darn big chromosomes. We have to go through a lot of cell divisions in our lifetime, however, and there is actually a limit to how long we can reduce the ends of chromosomes until we begin to delete essential genes. Once essential genes start to get deleted, that cell is going to go into something called senescence, so it's not going to be able to continue dividing. And this chromosome shortening that happens due to this replication problem is actually thought to be a big contribution in the aging process. Now, if you think about it, there are certain cells that have to divide a lot more than other cells. And so certain cells, like the gametes, have to actually produce the next generation. And we really cannot allow those chromosomes to shorten and shorten, and each subsequent generation has a shorter and shorter chromosome with less genes. Another kind of cell that is really sensitive to this problem would be stem cells, which continue to divide throughout the lifetime of an organism, making new cells to replenish the different tissues. Also, cancer cells, we don't like them to do this, but they will in fact divide pretty much indefinitely. And all of these cells have something in common, that they have come up with a solution to the problem of chromosome loss at the telomeres. They all express an enzyme called telomerase. So I'd like to tell you next about how telomerase works. But first of all, before we go into the details, I want to tell you that telomerase does not actually solve the problem of this overhang. We're going to start out here with this gap. We cannot make the second strand of that region at the telomere, and we're going to end up with the very same problem of a gap. However, the workaround solution is that we can actually extend the ends of chromosomes so that we prevent the loss of important genes. The way this works is via this enzyme telomerase. Telomerase is a multi-subunit protein, and it also has an RNA as part of that enzyme complex. The RNA that's part of the telomerase is shown in green here, and what it does is it base pairs with the bases in the telomere of each chromosome. That creates a template so that the top strand can actually be extended. And so here we extend that a little bit. And then what's going to happen next is that telomerase is going to ratchet over and go back from this, go back to do this again. And it will ratchet over and provide a little bit more template to, again, extend that top strand, or the old template strand in this case. Once we do that a number of times, that top strand has become much, much longer. And then at that point, the normal replication machinery can come in, make another several Okazaki fragments, and we've now lengthened the ends, and therefore we've prevented the loss of important genes. I want to end up by mentioning that there is a class of human disorders that are disorders of premature aging, and these are called progerias. And this is actually a 15-year-old boy. He looks like an old man because he has undergone this premature aging. What we know is that there are mutations in telomerase itself that can cause progeria, and also many individuals who have progeria, even if we don't know the cause, nevertheless they have really shortened telomeres.